So let us start with today's lesson of germination of seeds part 2. Now the learning objectives of today's lesson is how or what we are going to learn at the end of this today's lesson. So the essential environmental conditions for seed germination and the changes between what are taking place between seed germination, we should know that. So at the end of this lesson, we will be knowing this. We will be able to distinguish between the hypogeal and epigeal seed germination. So first we will learn what is hypogeal and what is epigeal seed germination. Now I think in the previous classes, like when you were in 6th or 7th standard, you have already gone through this hypogeal and epigeal germination. But now we will study this in details again. We will explain with several experiments that what are the conditions necessary and how they help the seeds to germinate. So seed germination with help of several experiments we are going to discuss, and that is very important for your exam, because we are drawing the diagram, we are taking the experiments to prove with several setups, different setups and different environmental conditions to show that how this seed germination occurs under certain suitable conditions. Okay. Now here, what is seed germination? So we all know that what is seed germination, but still we should see that what is the process. Now we have already studied that a seed is containing a dormant embryo in it, and that embryo is always in a state of inactive embryo. So it's not active. So how does it get activated? So dry seed, however, has several activities of life, including the exchange of oxygen and carbon dioxide. But all these activities are happening very, very slow within this embryo. So as this embryo is always in the dormant state in the dry seed, and the processes are going on very slow within very minute utilization of food when the seed is in embryo is in dormant state and seed is in dry condition. So is it clear? Now when this seed is having proper environmental conditions, is having necessary conditions for seed germination, it starts becoming active gradually. So this process, when the seed the formation of the seedling from the embryo is happening under certain environmental conditions, the process is called seed germination. So this process under several conditions or proper conditions which are necessary for seed germination, it is making the embryo active. So this process is called seed germination. Is it clear? So what is seed germination? The formation of a seedling from the embryo is called seed germination. So in one sentence, if we want to tell that the formation of seedling from the embryo is called seed germination, and previously when it was in dry state, some changes were happening within the seed also, but very slowly those changes were happening, and carbon dioxide and oxygen exchange was also happening, but they were having like very slow process. Now this, it is actually enhanced all the conditions when there are suitable environmental conditions and the seedling is being formed from the embryo, and this process of transformation of a seedling from the embryo is called seed germination. Is it clear? So let us see what are the types of seed germination now. Now what are the conditions which are necessary for seed germination? There are several conditions which makes the this seed germination favorable. So what are the conditions? Now first we should make a note of water. So water, what is it doing? Now most of the seeds are very dry and they must always absorb water for this process of germination to germinate. So water is absorbed, and when the water is being absorbed, what it is doing? It is softening the seed coat. And this softening of the seed coat, what it is doing? It is activating the enzyme, and that in turn is converting the starch in the cotyledons or the endosperms into simple sugar. So this absorption of water in turn, it softens the seed coat and activates the enzyme that converts the starch which was present in cotyledons as well as the endosperm, that is the specific tissue for food storage, into simple sugar. Now these sugars are providing energy for the embryo to grow. So what they are doing? They are providing energy for the embryo to grow. Now the next condition is, now here when you are telling about water, when you soak a seed in water, you can see that like you are soaking it at night, in the morning you can see that the seed swells up as it is absorbing the water, and at times the seed coat also ruptures. And generally after keeping it for a more time, what we will see that the radical elongates and comes out and forms the root system, and the upper part, the plumule, will form the shoot system. Now next condition is the air. So air is containing oxygen, and oxygen is always necessary for this germination process. What it is doing actually? Now rapid cell division occurs during this process of germination, and which helps in growth. And this energy is provided by the oxidation or breaking down of the food through respiration. So this breaking down happens only in presence of oxygen. Now as the embryo it begins to grow gradually, what is happening? The seed it cracks open and the seed coat has softened now. Now what is happening now? If the oxygen supply is not at all proper at this stage, what will happen? The seeds will not germinate at all. And this is why seeds grown very deep into the soil, they do not germinate. So once you put the seed very deep into the soil, they are not getting proper water, proper oxygen, neither proper temperature. So they will not germinate properly. Now next comes suitable temperature. Now seeds will only grow if the temperature is suitable, that is within a certain range. Generally we tell that 25 to 35 degrees is the optimum temperature or the necessary temperature for seed development. A very low temperature inhibits the growth. So if the temperature is less than this, that is very low, that will inhibit the growth. And a very high temperature, this one very low, is inhibiting the growth, whereas very high temperature, what will it do? It will destroy the delicate tissues. So high temperature, what is it doing? It is destroying the delicate tissues. It is destroying the delicate tissues. Is it clear? So low temperature is inhibiting the growth, whereas high temperature is destroying the delicate tissues. Now we will move on to the types of germination. Now we come to the types of seed germination. So mainly there are two types of seed germination, the epigeal germination and the hypogeal. So in the previous classes also you have studied the same thing, that epigeal germination and hypogeal. So let us discuss this in details. Now here comes epigeal germination. So what is this, the epigeal germination? Now the region of axis below the cotyledons is the hypocotyle. So this is the cotyledon. So this cotyledon, just follow my labeling. Now this is the cotyledon, the region of axis, thus below that, this region is called the hypocotyle region. And the region of axis between the point of attachment of the cotyledons, so this is the point of attachment of the cotyledons, and the plumule is the epicotyle. So region between the point of attachment of the cotyledons and the plumule is the epicotyle. So just remember it and just label it in the diagram so that it will be easier for you to recall. And once you memorize it, you will not be able to recall it during your exam. So just see the diagram and just try to memorize in that way, that the region between the cotyledon and this part is the hypocotyle, and between the attachment of the cotyledons and the plumule is the epicotyle. Now what is happening? If the hypocotyle elongates in this case, it comes above the soil and the cotyledons are always pushed above the ground. So if the hypocotyle is elongating, what is happening? It is elongating and the cotyledons are pushed above the ground. The cotyledons is being pushed above the ground and above the soil, sorry. This is called the epigeal germination. Now what are the examples of epigeal germination? The examples are castor, bean and cotton. The main three examples are castor, bean and cotton are the examples of epigeal germination. Now here you can see the cotyledons are being pushed above the ground and the hypocotyle is elongating. So the main thing happening here, the hypocotyle is elongating and the cotyledons are pushed above the ground. So till now, if you are having any queries, any doubts, please do write in my comment box so that I can clarify your doubts in the next upcoming video. So let us move on to the next type of germination, that is the hypogeal germination. So what is happening here in this hypogeal germination? Now the epicotyle, in the previous one the hypocotyle was elongating, here the epicotyle elongates. And the cotyledons, you can see they remain below the soil only. The plumule is carried at the tip. As the tip is elongated, the epicotyle is elongating. So you can see here the epicotyle is elongating here and the cotyledons are here below the soil only, and this is called the hypogeal germination. The examples are pea, gram, maize, rice and mango are the examples of hypogeal germination. And the cotyledon always they remain underground and the epicotyle is elongating. So how we write the difference between hypogeal and epigeal? So in the next slide we'll write the difference. Let us see. Now, difference between epigeal and hypogeal. What is the difference between epigeal and hypogeal? In epigeal germination, the cotyledons are pushed above the ground. In hypogeal germination, the cotyledons they remain underground. Now, in epigeal germination, what is happening? The hypocotyle elongates, and in the hypogeal germination, the epicotyle elongates. So this one is epigeal and this one is hypogeal. So this one is the epigeal germination and this one is the hypogeal germination. Okay. In the hypogeal, the epicotyle elongates, and in the epigeal, the hypocotyle elongates. Okay. And this can be seen in epigeal, can be seen in castor, bean and cotton, whereas hypogeal can be seen in pea, gram, maize, etc. So these are the major differences between epigeal and hypogeal germination. Now we are going to discuss germination with few experiments which are very important experiments. Might be they give you to write the experiment with a label diagram, or they can give you the label diagram of this experiment and tell you to explain the experiment in your exam. So either and or they can come like that. So just let us study those experiments. Now here the first experiment to prove that water is necessary or for seed germination. So what we have done, we have taken two petri dishes. Here you can see this petri dish A and petri dish B. What we have done here, we have placed equal number of seeds like green gram in both the petri dishes. What we have done, we have taken we have placed wet cotton wool in petri dish B, whereas in A there are dry cotton. So the cotton is dry. So we are here to prove that water is necessary for germination. So what we will see, that if we are keeping these two petri dishes at room temperature, both A and B, after 2-3 days we'll observe that the petri dish A, these seeds will not germinate, whereas the petri dish B seeds will germinate as they were placed on wet cotton. So hence it is proved that in the first petri dish, as no water was there, seeds don't germinate, and in the second one wet water cotton wool was there, so the seeds germinate. So hence water is necessary for seed germination. So this experiment is proof that water is necessary for seed germination. Now let us see the next one. Now here we come up with the second experiment where we are going to prove that oxygen is necessary for seed germination. Now we have taken two conical flasks here, A and B. So there we have put wet cotton wool in both the flasks, and we have placed equal number of soaked gram seeds in both. Now what we have done in this first conical flask, we have taken pyrogallic acid that absorbs oxygen, and we have hanged that test tube from the mouth of the bottle of this conical flask, and we have sealed the flask. Now in the second one we have taken water and we have hanged this near the test tube in flask B, and in flask B also we have sealed the mouth. Now in the first test tube A as well as B, both the test tubes we have sealed the mouth, and what has happened here, let us see. Now we have kept both the flasks at normal temperature, in a normal room temperature. Now after 2 days what has happened, we are observing that the seeds in flask B, they grow. So seeds in flask B, they grow, but the seeds in flask A, they do not grow. So they cannot grow. Why? They are not growing. No germination, they do not grow. So they are not growing. Why? Because this pyrogallic acid, what is it doing? It is absorbing the oxygen present in the test tube or the conical flask present within the flask. It is absorbing this pyrogallic acid is absorbing the oxygen. So no oxygen is present in this conical flask, and so no seeds are growing. But in conical flask B, as oxygen is present inside it, and as in the test tube water was there, so the necessary oxygen required for germination of the seeds was present in that, and so the seeds grew, or they started germinating in this second conical flask. So hence it is proved that oxygen is also necessary for seed germination. So this is proved. Now hence let us move on to the third experiment. Now in this experiment, we are here to show that optimum temperature is necessary for seed germination. So what we are going to do, we are going to take three different beakers. Now suppose we are taking three different beakers and we are labeling them as A, B and C. So like the same thing we have done in earlier experiments also, we are placing wet cotton wool in all three beakers. We are placing wet cotton wool in all three beakers with the green gram seeds inside them. So we are placing few green grams seeds inside them in the wet cotton wool, and what we are doing now, we are testing the temperature, that optimum temperature is necessary for seed germination. So what we are going to do, we are keeping the first A at room temperature. So A beaker is being kept at room temperature. Then we are putting the this beaker in the refrigerator. So it is in a cool temperature we are keeping. And we are keeping the C1 in heated oven. So we are keeping the third one in heated oven. So first one at room temperature, second one in refrigerator, and third one in heated oven. So what we are observing, so after 3 days, we will observe the seeds. We will take out the beaker from the refrigerator, we will take out the beaker from the heated oven, and we will see the seeds in the wet cotton wool under room temperature. So what we will observe? We will observe that the seeds which were under this room temperature, they have germinated properly because they had got the optimum temperature. So as we have discussed earlier, the optimum temperature is from 25 degrees centigrade to 35 degrees centigrade. But when we are putting it inside the refrigerator, the temperature will be 0 degree to 4 degrees centigrade. Once we are putting it in a heated oven, it will exceed like 100 or 200 degrees centigrade. So this either too high is not adequate for this germination, and too low is also not at all adequate. So there should be an optimum temperature where only the seeds will germinate properly. So in this experiment, we will say that the seeds in a this flask only will germinate properly. So this is the experiment which is showing us that the optimum temperature is always necessary for seed germination. And in the next slide, we will be discussing that how the three conditions are together. They are acting together for the growth of a seed, that is water, temperature and oxygen. So let us move on to the next one. Now in this third experiment, what we are going to show? We are going to show that both water, temperature and oxygen are necessary for seed germination. So first we have taken water, then we have already taken oxygen. Now in this we are taking the three conditions only: water, temperature and oxygen. All are required for seed germination. So what we have taken here? We have taken a glass slide. You can see we have taken a glass slide here with the help of a thread. We have taken three air dried bean seeds here at three positions. We have labeled these three seeds as A, B and C. Suppose we are labeling the seeds as A, B and C, and we have stuck them in a slide. So we are keeping the slide in a beaker containing half of water, and the top seed we are telling that it is the C, and middle one is B and the down one is A. And we are keeping the whole experimental setup in a warm place with adequate light and temperature. Now what all are required for this germination process, we have kept under that only. So what we are observing here, so after 2-3 days, what is our observation? We are observing here what is happening here, that the middle seed B is germinating. So the B seed, it's germinating. Okay, because the B seed, it is having adequate oxygen, it is having adequate water, as well as it is in optimal temperature. Whereas the seed which is in the air, that is the C1, C don't germinate because it is not getting water, not getting adequate water or moisture, not getting water. Now the A1, why it is not germinating? This is also not getting what it is not getting. This one, the top one is not getting oxygen. It's getting oxygen and no water it is getting. Whereas the A1, it is getting only water. This one is getting only water, and it's not getting, no adequate oxygen is there. So no adequate oxygen is there, only water it is getting, and three conditions which are necessary for germination are not present in A and C. So A and C, they don't grow properly, whereas the B, which is getting adequate temperature, water and oxygen in adequate amount, it's germinating properly. So these are the three main conditions necessary for germination, that is water, temperature and oxygen. So now let us move on to the next one, where we will see that how the seed germinates under optimum temperature. Now here, just for your knowledge, I'm discussing about a different type of germination, that is viviparous germination. So this viviparous germination, what is it? Now generally in mangrove plants like Rhizophora, what is happening? The seed is germinating inside the fruit itself. So this is a speciality where the seeds are germinating inside the fruit and while it is still attached to the plant. So when it is still attached to the plants, the seed is still germinating inside the fruit only. And what happens after germination? After the germination process is completed, the seedling, they drop down into the soil and then it grows to root and gradually it develops into a plant. So after the seed, the germination is complete within the fruit, the seedling drops down on the soil and it develops into roots and gradually it grows. So this type of germination is called the viviparous germination. So this is an exceptional thing which is different from hypogeal or epigeal germination, and this is a kind of germination which is happening in the mangrove plants or the plants which are generally in mangrove origin. So this we have already discussed, hypogeal and epigeal. This is the third kind of germination, viviparous germination. Just remember it in case if you are getting this different type of germination in your exams. So this happens only in mangrove trees. As of now, I've completed this full lesson of germination in seeds. So please go through the first part. I am putting it in the link above as well as in the description box also. And if you have not yet gone through the previous video, please go through it. It will make your studying easier for this lesson. And if you're not yet subscribed to my channel, please do subscribe to my channel and don't forget to like and share my videos. And stay tuned, stay updated with my YouTube channel. Thank you.